Hello my dear students, welcome to my channel Online Biology Tutorials. In this video we will study about a dreadful disease which is named as cancer. So cancer basically happens because of uncontrolled cell division. Generally, when mitosis becomes faulty, means abnormal mitosis gives rise to cancer. We already know that metaphase occurs using these four phases: prophase, metaphase, then happens our anaphase, and then in the last telophase happens, whereby just after this cytokinesis happens. So just when prophase is completed and cell enters metaphase, there are checkpoints in between. These checkpoints are actually the factors or the protein that helps in initiating the next stage. Means a checkpoint will be there between prophase and metaphase, and it will check that whether prophase has been accomplished right or wrong. Similarly, checkpoint will be present between metaphase and the anaphase, and this checkpoint will check are all the chromosomes arranged on the equator. If is it so, then it will give a signal that now anaphase may happen. Similarly, if anaphase is accomplished properly, then a checkpoint will move this cell in the next phase. Thereafter, when the telophase will get over, if it has happened correctly, the cell will do cytokinesis, and ultimately from one parent cell, how many daughter cells will be formed? Yes, 2 daughter cells will be formed if it is the case of the normal mitosis. But when these checkpoints, they become faulty, they become mutated, because these are nothing new, these are made up of the proteins. So when proteins, they get mutated, of course the checkpoints will become faulty. In this case, prophase has not accomplished completely and the cell has entered metaphase. All the chromosomes have not been arranged on the equator and they have moved to different poles. So that means unequal chromosomes may reach at the opposite poles, and this may lead to the occurrence of more than 2 daughter cells. And this disease is known as cancer, wherein the uncontrolled cell division may happen. This is the key word for this disease. Now, the agents that are responsible for giving rise to cancer, they are known as carcinogens. Okay, so carcinogens, they are the agents of cancer. And if you talk about the types of cancer, any uncontrolled division is known as tumor. Okay, so if the number of cells, they are dividing abruptly, tumor may be seen. It could be benign tumor, or it could be a malignant one. Now, benign tumor, they are the one which are restricted at a particular place. Means if uncontrolled cell division has happened, but these group of cells, faulty cells, which are formed because of the faulty process of mitosis, they will be restricted. So when they will be restricted to a particular place, we will be using the word benign. Whereas malignant word will be used when the cells, they are going to transfer from one place to another. Right? So if certain area has become cancerous, these cells may come in the blood, and from the blood, because blood is circulated in the body, so these faulty cells may transfer a normal or a part, and that part will also become cancerous. So this property of getting transferred to the healthy part is known as metastasis. So malignant tumors have the ability to invade a healthy part. They show the property of metastasis, whereby the healthy cells, they also get affected. Okay, so carcinogens are the agent, benign tumors are restricted to a place, malignant are the ones that have the capacity to move from one place to another because they show metastasis. This term is important. Now, what are the various agents which are responsible for giving birth to this disease, right? So, of course, exposure to UV rays, X-rays is harmful and it may give rise to the cancer. Fine. And there are viruses which are also responsible for giving rise to the cancer. Such viruses are known as oncogenic viruses. Okay. So oncogenic means they are cancer causing. Human beings also contain proto-oncogenes, and when these genes become active, then a person suffers from cancer. So these are the few reasons which are mentioned in your book and you need to learn them. Exposure to the UV rays or harmful radiations may cause the cancer. Oncogenic viruses, if they invade the body of a healthy human being, then also cancer may happen. Proto-oncogenes, if they are becoming active, then also cancer will happen. However, sometimes it has also been seen that replication errors also give rise to the cancer. Cancer is a dreadful disease. Actually, the stage in which it is diagnosed, it is very, very important. Generally, there are 4 stages of cancer: stage 1, 2, 3, and the 4. 4th stage is the one in which death happens. So if a person is diagnosed with cancer in stage 1, there is a probability of giving a cure, right? So if we want to treat the cancerous patient, there are few measures which are available. So treatment is our next heading, and in the treatment, surgery can be given, or radiotherapy can also be given, or immunotherapy can also be given. We'll see what happens in each therapy. Surgery means the affected part or the place where cancer is there, that part will be taken out with the help of surgery. Fine. In radiotherapy, harmful cells or the cells that have cancer or that the cells which are mutated, these are exposed to the radiations, and these radiations will kill the affected cells. Okay, so by radiotherapy, radiations are given to the affected cells and these cells are being killed. In the immunotherapy, chemotherapy is given. Chemotherapy is a special type of treatment which is given to the cancerous patient, which includes killing the cancerous cell also, but it kills non-cancerous cells also. In chemotherapy, the follicle cells of the body also gets damaged. So hair fall is seen in the people who undergo chemotherapy, and this is a good indicator. It says that the medicine is effective, that's why hair follicle, they are getting killed. That's why the people who take it, they generally become bald. But over a period of time, hair follicle regrows and hair growth happens again. Generally, when a person suffers from cancer, mixture of these 3 therapies is being given. Means surgery is done and then radiotherapy is given in various cycles, means one after the other. After a certain time span in between which will kill the affected cells, and chemotherapy is also given. Generally, if the cancer is in the advanced stage, doctors generally don't prefer to give surgery, because it is said that as scissors touches the affected area, it actually acts as a stimulus and it helps in increasing the cancerous cells to an enormous rate. So surgery is only given in the initial stages when cancer is diagnosed in the early stage. In the subsequent stages, in stage number 2 and stage number 3, generally surgery is not preferred. At that time, radiotherapy and immunotherapies are given. Now, what are the various diagnostic tools that will help in determining that a person is suffering from cancer or not? So we will study the diagnostic test for it. Very important one is the biopsy. In biopsy, it's a tissue specific test. Okay, a tissue is taken. This question was asked in one of the exams. Is it a cell specific test or tissue specific? Because we take group of cells, and group of cells, you know, that we call them as tissue. That's why it is a tissue specific test. So in biopsy, the suspected area where cancer is expected, tissue is being taken and it is grown in the culture, means it is grown in the nutrient medium. Now see, very important property of the normal cells of our body, that they show contact inhibition. Contact inhibition means as soon as they come in contact with the surface, they stop dividing. Okay, but cancerous cells, they don't show this property of contact inhibition, and that's why it can be tested through a biopsy test. Suppose a tissue is taken and this tissue is grown in a nutrient medium. That means we have cultured it for few days. Now, as soon as these cells will come in contact with the surface, they will not grow if these cells are normal cells. On the other hand, if the cells are cancerous, then they will keep on dividing because they will not follow contact inhibition. They will form pile of cells because they, when get in contact, they will keep on dividing. So here you can see that the cells are continuously dividing, and getting touched with each other is not actually stopping their division process. So these type of cells that make heap of cells in the culture, because they are not at all following contact inhibition, they are known as the cancerous cells. So cells are grown for a couple of days, and then when reports come, they actually define that whether the person is suffering from cancer or not. Other two tests which are used for detecting cancer are CT scans. CT scans are computed tomography. These are the special scans which are done on the CT machine. There is one more machine which is known as MRI machine that does magnetic resonance imaging. These two are the specific tests for the cancerous patient. So you need to learn all these three rather. Contact inhibition is also very important. Biopsy is a tissue specific test where the tissue is grown for a couple of days to notice its growing pattern. If it is forming a pile of cells, the cells are cancerous. But if only one layer of cells is seen, that means they are following contact inhibition, and such cells are not cancerous. And CT scans and MRIs, they are also used for diagnosis of the cancer. Hope this was helpful to you. Stay tuned for more videos and don't forget to subscribe my channel among your family and friends. Happy learning!